great pleasure to introduce the R&D panel, which will be led or moderated by Martin Mackay, who's the co-founder of Rallybio and quite frankly, very experienced in the field, been in every section of our business. And joining him, and the reason that I'm introducing the panel is Andy Plump, our great moderator for the whole thing, and the president of research and development at Takeda. Joining him will be David Reese, who's the executive vice president of research and development at Amgen. And I believe John Reed will not be able to join us today due to some travel difficulties. And also Mathai Mammen, who's the executive vice president of R&D at J&J. And it's really a great pleasure to see you guys again. And over to you, Martin.
Thank you very much, Deval. It's great to see you, albeit on screen. Hopefully, in a wee while, we'll all be together in person. Before I start this panel, I was just going to say to Karun and Andy how magnificent the day has been. I think the panels have been truly terrific. Great panelists, great questions, a range of answers, and the polling was also fun. And I think, Andy, you've taken it one step further now, actually giving blood for this meeting. I mean, that's really extended it. I was rather worried this morning when I saw the Band-Aid, but we understand how that happened. It's unfortunate that John can't join us. He's had a genuine travel problem today. No matter, I think we will continue with three marvelous panelists. I actually got some feedback last year. As you remember, I went straight into the questions, and I got some feedback that not everybody knows Andy, Mathai, and David. I just assumed that, you know, they're some of the most famous people in our industry. So before I get into the questions, I'm just going to ask you just to give a brief introduction, really focusing on what you're doing just now in your respective companies. Then we'll launch into questions. And then we already have some questions from the audience, and I'll go to those so that we get a really good feel for an interactive session. So Mathai, maybe you can kick us off with a brief introduction.
Sure. Mathai Mammen. Born in India, raised in Canada, educated in Massachusetts, started a company called Theravance in California. From there, we did some great things. Proud of that team. Proud of the company, moved on to Merck, and then now for the last 5 years have had the privilege of running the R&D group at Janssen.
Thank you, Mathai. David?
Hi, Dave Reese. I'm the head of research and development at Amgen, a product of small town middle America, very tiny town middle America. Have been at Amgen for 17 years in a variety of roles. Trained as a medical oncologist by way of background. This is the only company I've been, so I voted with my feet for 17 years now.
Thank you very much, David. And Andy.
Hi, Martin. It's good to see you, all of you. I'm Andy Plump. I'm the head of R&D at Takeda Pharmaceuticals, and weekends I do a little bit of sparring, boxing in the ring, and that's a left uppercut to the chin. That's how I got the injury, just to set the record straight. I've been at Takeda now for about 8 years. And in fact, when I started this job, I was so intimidated by the scope of the job. You might remember, Martin, I reached out to a few former R&D heads, of which you were one, and you were so gracious in bringing me in and telling me, Andy, it's really fun, but it's really hard. And I can tell you 8 years in, it's been both. We are focusing on really taking a company that had a 240-year history predominantly in Japan and really globalizing the company. And we've really done a lot of work over the last half decade plus to change fundamentally the Takeda R&D organization. And we're kind of halfway into that journey right now.
Marvelous, Andy. Thank you. And I remember that session very well indeed, actually. As I was preparing for the session, I was really just thinking about, and I think about just the critical importance of what you folks are doing. And if it's individual companies, it's quite amazing. But if we put it together into the ecosystem of pharmaceutical R&D, you're carrying so much weight on your shoulders. And I have to say, you always carry it lightly, at least in public, to great effect, actually. I'm going to start with a philosophical question. And it's one that we've touched on before. It has a subpart to the question, but I'll concentrate on the main part, which is, what are your feelings now in terms of where do you think we are in the evolution of therapies, from the incremental benefits that have been so important across so many diseases through to truly now transformative therapies, and always approaching that element of functional cures? I mean, this is what we'd all like to do. So in that spectrum of where we've been, where do you think we are, and what's going to take us to truly get in the functional cures? Let me start with you, David.
Yeah, maybe I'll start at a very high altitude. To me, this is, what I like to call a hinge moment. I think we will look back in a decade or two and say that this was the moment where things in our industry really started to change. What's propelling that change? To me, it's the uniting tech and biotech, in a way that's qualitatively different, that will, drive insights into biology and drive, what we're now seeing is just an absolute proliferation of therapeutic modalities, more than one can keep track of, certainly far more than any single company can be expert in and can focus on strategically. So to me, this is the moment. We're seeing examples play out in real time. It was just almost exactly a year ago that DeepMind deposited 350,000 putative protein structures in an open access database overnight. That's a before and after moment. That's a before and after moment. We started downloading structures and code within hours and started working with it. So there are a lot of elements here, but to me, at the highest level, this is the moment. And the question is, as an organization, where do you focus? Where do you focus your investments, and how do you then capitalize on this union of tech and biotech?
Oh, that's marvelous, David. Thank you. Very good example there. Andy.
Well, I remember when I joined the industry in 2001, there was one focus in the biopharmaceutical world, and that was making drugs in primary care. So this whole idea of low volume, high price points and specialty care just really hadn't taken root. And there was no driver, there was no structure to incentivize this idea of transformative therapies and functional cures. We were all incentivized to develop next in class. And if you look back at all the different agents that we went after, we were all going after the same targets: calcium channel blockers, statins, angiotensin receptor blockers, PPIs. It was all the same target over and over again, all using the same modality. And it's just amazing what's happened over the last 2 decades. And the part about it that I find the most compelling is that, when you talk to anybody in the ecosystem, whether it's a scientist, whether it's a clinician working in development, whether it's your commercial colleague, whether it's a regulator, a payer, a patient, everybody wants the same thing, and that's innovation, drugs with very high effect sizes and the best, possible safety profile. I think that's terrific. Science has taken off. I agree with David fully. A large driver of our ability to now go after transformative therapies and functional cures is driven by the modality toolbox that we have access to. And now with emerging genetic therapies and cellular therapies, which have the potential to do the kinds of things routinely that are challenged by small molecules and recombinant proteins in general, it really sets us up. There are 3 challenges I see, 3 main challenges with tons of challenges. No one has to underestimate the challenge. But I see 3 main challenges in our ability to really inflect around our aspirations for these types of therapies. The first, and I'm sure everybody will agree, is just fundamentally our understanding of disease, right? There's still a gap there. Second is we have the technologies to modulate up or down any disease target, whether it's a protein target or a nucleic acid target, anything. The problem is tissue targeting. So homing that therapy to the right target tissue. And then the third is in our clinical trial space, having the tools and the endpoints to actually study the drugs that we develop. It's still a big challenge. Even fundamentals like identifying dose can be really difficult in certain areas, like in CNS disease. And then certainly clinical endpoints. We just came out of a panel for rare diseases. Many of the rare diseases are so insidious in their natural history. It's very difficult to understand what endpoints to look at and then to get agreements with regulators.
Yeah, great point, Sandy. I'll come back to the modality piece. As you say, just the tremendous progress that's been made, in the world that we came into this industry. And Mathai.
Yeah, so the question you're asking is what's pushing us towards transformative medicines or curative medicines and not so much to incremental medicines. And, I think it's the chances of getting to any destination are dependent to me on the alignment of forces. If there's like equally strong forces pushing in opposite directions, that's a problem. In this case, I'm really optimistic. It's because the major forces kind of all move us towards transformative therapies. Like, the science is definitely supportive. Like lots of investment around the world now, like from some regions that were not contributing cutting edge science. Now all over the world, there's cutting edge science feeding innovation. Second is, as Andy alluded to, some of the modalities on which to capitalize on that science and move things forward. That's clearly a force that's allowing things to bubble up. And third, even though everyone kind of complains about it, the pricing pressure, the payer pressures, what will be reimbursed, actually does raise the bar. So everyone's setting out in companies right now for making something head and shoulders more preferable for a patient than anything else they might be able to use. And so that sort of stringency in the system, even though it's an opposing force for any one company, for the industry as a whole, makes the industry as a whole head towards transformational and away from incremental. As Andy said, there are a ton of challenges, but those are major forces all driving us towards the destination I think that we're proud to go in.
Yeah, that last
point. I couldn't agree. Oh, please, David, please.
Yeah, last point's really a fundamental one, Mathai. In fact, as one of our 3 strategic imperatives at R&D at Amgen, we have included enabling access and use. That's something that an R&D organization wouldn't have really conceived of a decade ago, and certainly 2 decades ago. But we are not divorced from that tectonic plate that is pushing the industry.
Yeah, and I think it gets back to the very mission of why we're here. That a regulatory approval per se is at best a halftime show, and I know all the people on this panel are here because of the impact on patients, and that fundamentally requires access. So of course it should be part of a goal.
Yeah, without doubt, great comments from all of you. I'd like to stay with you, Mathai, for a moment and then go to the others. I see the juxtaposition here between moving to this functional cures, but you raised rightly the patients. And how do you think we're doing in terms of identifying patients earlier in disease? And then really importantly, a point I've heard you making, knowing which of those patients are going to progress in that disease. And then along the way, surrogate endpoints, can we get to earlier answers rather than waiting for the progression of a, dreadfully chronic condition? Where do you think we are in that loop now?
Yeah, no, thanks for those questions. Martin, if I can elevate just for a second and say, like, fundamental to our problem, one of the challenges is figuring out who to treat with what mechanism. And it's one of the challenges, I think, that goes into, it's part of the answer to your questions, but, it should bother us that a drug as great as a TNF blocker has sustained efficacy in maybe 10 or 20% of people that you treat. It should bother us, right? It should, because how do you know who they are ahead of time? Because everyone else might benefit very transiently or only have side effects. So part of our challenge as an industry is more mechanistically defining disease. And so that requires understanding disease. And hence, like, I like the answer to the last panel's question on what one of the big challenges of rare diseases, and it's understanding disease. That's true for common diseases as well. Like, really understanding it mechanistically, which is not a trivial task. And if you do that, then you can, for progressive diseases, things that get worse with time, like many do, then the earlier typically one identifies them and treats them, the better. So my answer to that is in part to take advantage of the large collections of longitudinal data that are increasingly available, and be a bit more humble in how one approaches them. So you don't need necessarily at t=0 to be able to draw a mechanism on a board in order to go after it, but rather have hypothesis-free generation of data points that allow for the creation of testable hypotheses, just like a more agnostic approach. And if you do that, then you can ask the question, in how you're identifying a subgroup, maybe 10% of people are rapidly progressing from first identifying them as type 2 diabetics to substantial sequelae of type 2 diabetes, or some group of people that have had a surgery that are at risk of thrombosis, only some percentage of them are particularly prone, and why. So at first you may be biased and understand and look for kind of the obvious stuff under the lamppost. But I think allowing one to just swim in the longitudinal data might identify different ways of defining those cohorts that say, okay, it's this piece of biology ultimately that we identify, and it's that is then the disease. Let's call that the disease. So that helps. Like, the understanding of the disease is foundational. The allowing yourself to be influenced by longitudinal data and where that points you is second. And then the third is being brave about, you asked it, Martin, about regulatory endpoints and how far ahead. You have to be brave about knowing that you're in an area that's important, presumably that you're in it because it's important, for a long time. Like, it's very rare that you come in and be in and out in 5 years of any disease. So allow yourself, when you go in, to start the hard work of identifying early reads, like biomarkers, so to speak, that might ultimately predict an outcome that's obvious to the public, like death or hospitalization. But what are those biomarkers along the way? As all of us that have been around biomarkers for a long time know, it's a long path. And so when it's an important area, I think it's important for maybe individual companies and maybe consortia to begin that path to say, what is it that is an early enough read that we can get there and develop more medicines earlier in the course of that disease?
Great points, Mathai. Really terrific thoughts. Andy, can you continue with this theme, please?
I will. I'm going to take it in a slightly different direction, but I want to just underscore a couple of words that Mathai used: humility and brave. He said both of those words. And I find in this business, the more I'm in it, the more humble I become. And what kills great opportunities is arrogance in our business. And I do think we need to be brave in pursuing our hypotheses, because so much of what we do fails and we have to remain so resilient. So where I had interpreted your question is, as an industry, how are we doing in terms of treating disease earlier and earlier and moving more into early stage disease and even preventive diseases? And I'm such an optimist. You give me a glass, no matter how empty it is, it's that percent full. Okay, I'm a 10% full glass kind of guy. And I'm sure that Mathai and David and you, Martin, are the same. Otherwise, we couldn't be doing our jobs.
Yeah, exactly.
But this is an area where I don't have much optimism, actually, because if you look at the kinds of therapies that we're trying to develop, they're very hard to study in settings in earlier disease, and certainly almost impossible to study in preventive settings. If you look at cancer, for example, almost every time we go after a new cancer, we go after patients who have the most severe forms of disease, relapsed and refractory forms of disease. Now, there's an incentive to move up in lines of therapy in oncology, but it's really hard to do, especially as we have standards of care that are okay. because your comparators drive out longer and longer trial times. Even look at a disease like Alzheimer's disease, which has been perhaps the hardest nut for us to crack as an industry in terms of a large disease. We are moving up into more kind of mild and early forms of dementia. That may not be early enough. There's some speculation that you'll need to go 10 or 15 years before disease onset, because that's when you start to see neuronal damage that you can't really read. That's a very hard study to enroll. And then, there's an issue around incentives that's built into our exclusivity system, where our goal is to get great medicines out, but then those medicines need to drive a return that can then feed back our investments. And if you get a drug approved in a refractory setting and then it takes 10 or 12 or 15 years to study it in earlier settings, that's just not possible. So somehow there needs to be some bridge to what we do, bringing forward these innovative therapies, to then, as a society, finding ways to study them in earlier forms of disease and in preventive settings.
Yeah, no, great point, Sandy. I'll just make a comment at this point before going on to you, David. It's quite interesting. Having worked now in rare disease for 9 years, almost a decade, you certainly learned very early that you have to be humble. There's no question there. And actually in the diseases that we worked in, you have to also go very deep. There's so few patients often involved. There's a real depth. I think about our understanding of paroxysmal nocturnal hemoglobinuria, or hypophosphatasia. We had to go deep. But interestingly, just as a very quick anecdote, one of our programs in the company that we run, called Rallybio, one of the lead programs is completely preventative. Now, it's a little unusual. You don't often get this chance, but there's a mismatch between platelets of the mother and the fetus. Think Reese's disease of the newborn. It's the platelet equivalent of that. And here you have a situation, if you think about it, the mother's healthy and the fetus is healthy. It's just, this mismatch that causes a problem. So our answer to this, we hope, will be a prevention, complete prevention. They will continue to be very healthy. That has taken quite a lot to explain to people that it's actually a prevention. Folks in the ecosystem like there to be a patient that's ill and you do something about. So it's just an added complexity to some of the things that you're both raising. Let me pass to you now, David, for your thoughts on this area.
Yeah, let me provide a third angle, as it were, on this question. One of my beliefs is that we've entered what we call the era of human data. One thing that Mathai and Andy never mentioned, nor did you, Martin, was animal models, better preclinical models. Everything we were talking about was, in fact, human data. And I like to define human data expansively. So multi-omic profiling now of disease, the increasingly rich clinical trials data that we've got, safety, efficacy, biomarkers, and then, of course, the tsunami of real-world evidence and real-world data. This is where, of course, much of the longitudinal data that Mathai was discussing, that's where it's derived from. A few weeks ago, I had the pleasure of going to Iceland, to Reykjavik. We celebrated the 25th anniversary of Decode Genetics. Decode started, of course, with genomics. When Amgen acquired Decode, they'd done a couple thousand whole genomes, had tens of thousands of individuals who were genotyped. Now there are 2.5 million individuals that are genotyped, hundreds of thousands on whom we've got whole genomes, and many tens of thousands now where we have transcriptomic and proteomic data. So you have these enormously rich data sets to not only define disease risk, but I think, just as importantly, two things: disease progression, which you mentioned, Martin, because often the molecular profile for risk and progression are not the same, and we need to understand those if we're going to intercept the disease appropriately, and then to really determine subsets of disease. I think one of our challenges in many diseases is that there are, in fact, multiple diseases. We know in oncology that breast cancer and lymphoma, for example, on a molecular level can be very discretely segregated into specific diseases. Well, we know the same thing must be true for diseases like lupus. Why do some patients have kidney predominant disease? Others have skin only disease and or joint disease. It's clear that there are, molecular subsets of that disorder. We just don't know what they are yet. And I don't think genetics alone will get us there. So I think it's this sort of broader profiling, longitudinal use of those data that's going to drive the biologic insights. And in part, this reflects back to this union of tech and biotech, because you need tech to actually make tractable these data sets. I'll end with what I think is kind of an astounding fact. At Decode now, we have almost 200 petabytes of data across these data sets. Simply organizing and getting ready to manipulate and analyze those data is a challenge in and of itself. It's, of course, been famously said that quantity has a quality all of its own. And when you're talking about that much data, it absolutely has a quality all of its own. It is, an enormous opportunity and an enormous danger, because as these data sets become publicly available, anyone with half a brain and a statistical program is going to uncover a lot of correlations. And what I always tell my team, of course, is what I'm really interested in are the ones that are true. And understanding which ones are true is a much trickier business, but tremendous, amount of excitement about the potential for human data to really propel this field.
Can I add one point to that very important set of statements? And, you said it, David, and, yourself and Amgen and with Decode and Regeneron and the Broad and pioneers, and now many companies and institutions have really become powerful at genetics and at both the DNA level and the transcriptomic level. And you said it, that you're adding to that right now other data sets. And I think it's this fundamental notion, if you step back, of we have a hypothesis that there is a certain amount of nature that is going to predispose you to illness. But the measurements of all the consequences of nurture aren't as talked about. Like, what is it that describes us at our age on this panel right now? How do we best describe our non-genetic state? Is it full characterization of our immune system? Is it our proteomic, descriptors? Is it, can we get to finally to metabolomics at such a rapid rate that's going to be practical? So, we do welcome and need new technologies that make rapid, just like DNA sequencing allowed it, to characterize the nurtured state better and better and better, to understand disease categories, propensity, and so forth. And I think it is a matter of where the technology is at the moment.
In fact, the line is blurred.
Yes.
For example, Decode has published on this. A patient who develops lung cancer as a result of smoking, it's an environmentally driven disease, of course, a known risk factor. But in fact, individuals have a much higher propensity or likelihood to become addicted to nicotine, which is genetically driven. So is that now a genetic disease or an environmental disease? Well, the answer is, of course, is both. Yeah. And it's a very complex interplay.
No, absolutely, David. I'm pleased that you're also mentioning Decode. I thought that was a very brave collaboration that you did at the time. I think most people did, and clearly it's bearing fruit now. So well done. I've got lots more questions, but questions are now coming in the chat line. They're coming in. I'm delighted to say that we have online now Dr. Shiv Sarin. Shiv is the Vice Chancellor of the Institute of Liver and Biliary Sciences, and he actually joined us at this panel last year, but unfortunately it was in the middle of the night in India, and I'm delighted to say that he's actually calling in from London today. So Shiv, may I go to you for your question for this panel?
Well, first, very nice to see Andrew and Mathai and everyone, and David and you. And of course, thanks to Karan for inviting. I was just going to follow up on the trial designs that you have. 1/3 of the world has fatty liver, and the drugs, they work differently. And I think at least in the London European meeting on liver, all of us were thinking whether we should not stratify our inclusion criteria based on several genes, because at the gross level, it looks like every fatty liver is same, but there may be 100 variations in that. Some of them are rapid fibrosers, some are slow fibrosers. So my question is, and are the pharma companies thinking of starting with trials, large-scale trials, where the genetic data is also available? And if so, how do we define our endpoints? I thought it might be worth its while, because it's very hot right now in the scientific, at least liver scientific world.
Thank you, Shiv. Andy, maybe you could kick off.
I want to thank you for calling on me, because I was going to jump in even if you didn't call me, Martin. Shiv, it's good to see you, and thank God that you're in London, although it is not close to 9 o'clock on Friday night in London. Actually, we're working on a liver disease. It's a rare form of NASH called alpha-1 antitrypsin-associated liver disease. And actually, we'll be coming out, it's timely to ask the question, we're coming out with a paper in the New England Journal this weekend with our partners at Arrowhead. And we have an RNAi, RNAi that knocks down the mutant form of alpha-1 antitrypsin that forms these toxic aggregates that are pathogenic to disease. Now, that's the kind of genetics that I like, because it's just so clear. And then you'll see this in the New England Journal paper, even when you have something that's just so obvious and so clear in terms of knockdown of a pathogenic causing gene, proximal biomarkers that you can use to define dose, and even intermediate biomarkers that can help you understand what's happening in the liver. Even in those settings, the regulatory agencies are really challenging to work with in terms of endpoints that are necessary for registration. And that's in a disease where the pathogenesis is fairly well defined. I think as you start, that's how I like to approach common diseases, is start with a really well defined genetic subset that you can use as a stepping stone to start to make correlations between proximal biomarkers and disease endpoints. And then you can start to look at those proximal biomarkers in more heterogeneous diseases. I think in many disorders, let's take Alzheimer's as, again, a prototype, where you don't have those really, maybe you do in Alzheimer's disease with APP and amyloid beta mutations, but in many diseases, you don't have those genetic, and the prototype here, by the way, was familial hypercholesterolemia with LDL, where you could go very quickly from a rare disease to the common disease. I think in a lot of common diseases, we try hard to identify those segments that are going to respond to a mechanism, but it's very difficult if you don't have a target that attacks the genetic etiology that you're going after, to know whether you're going after the right segment. So sure, you could reduce that common disease down to a more homogeneous disease, but mechanistically, if you're not going at it with the right drug, as I think we are with alpha-1 antitrypsin, it gets challenging. And so what we tend to do, as opposed to taking that risk, we'll go after the general population and then post hoc start to look in retroactively for those responder segments.
I may just add, maybe you have a winning game with alpha-1 antitrypsin. There was a paper long ago. Those who have a specific type of allele of alpha-1 antitrypsin, they get more alcohol-associated liver disease. So maybe I can send you that paper. That may be a winner for you. And you have a haplotype which with alpha-1 antitrypsin does not get liver disease despite drinking. So there is a basis, and alpha-1 antitrypsin is just not that disease. It's a very important gene for most of the liver secretory functions.
Yes. Yes.
So my question still remains, are the companies going to start with, taking inclusion criteria with phenotype plus genotypes rather than just phenotypes?
Let's maybe Mathai, you could make a comment, and I'll move on to some other questioners that we have. I'll come back to you, David. Mathai?
Sure. I think in general, the companies are certainly, we are trying to baseline profile in whatever way we think might be predictive or identify the right groupings of patients. So in some cases where there's some sense that there's a genetic driver, like certainly in cancers, in other some of the common diseases we have, we'll baseline characterize that way. But in other cases, it is a late onset something that there are clearly other things going on, and maybe it's an immune characterization. Maybe it's an immune characterization that's more important than germline genetics per se. And that might be the way we make sure we do that, as opposed to just clinical, descriptors. And so we'll try. It's quite circumstance specific what we'll baseline characterize.
3 gene
Thank
chips.
you, Mathai.
Yeah. 3 gene chips are there, PNPLA3 or the transmembrane and others. So if you have that, then you're likely to develop fatty liver much faster and transmit it also. So now people are thinking of using that group, block them and leave the rest so that the others are not included in trials, because they will be very, very slow. And the other group may develop cirrhosis maybe in 8 to 10 years. Anyway, these are my thoughts as a clinician. But I thought drug companies would be ahead of us and thinking of such pheno-geno combinations.
Thank you, Shiv, and great to see you this year.
Yes.
I'd like to go to another questioner from the audience, Saeed Kazmi, who I know from previous meetings, the CEO of Jubilant. Great to see you on screen, Saeed. Please answer your question to this panel.
Thanks, Martin, and thanks to Mathai and Dave and Andy for yet another very insightful panel. I wanted to pick up a little bit of where, Martin, you started. While there is a merit in the conventional wisdom of success breeds success, I'm just afraid that we may be seeing sort of a herd mentality when it comes to, pipeline at many pharmas, but more so in biotechs. So thanks and full credit to initial innovators. I mean, now we have close to over 80 CD19-based cell therapies. I mean, tens of RTK and KRAS inhibitors that are out there. And my business development colleagues tell me that this is, if you have those, pharma would be interested. I mean, last week at Bio, I met an ex-colleague of mine who is a senior executive at one of the large pharma companies that would remain nameless, and said something like, if you have any mRNA project, bring it on. We have lots of money. So I think my question to you all is, are we running the risk of just maybe wasting too much time, energy, and precious R&D dollars to chase incremental benefits, as Martin had initially mentioned, of proven MOAs? And what message would you have to innovative biotechs out there to take risk with new creative transformative therapies where, pharma is willing to share the risk and partner? I mean, am I better off developing a novel drug for brain metastasis as a biotech, or should I go for 7th or 9th SHIP inhibitor that I can partner to pharma overnight?
Thank you. Very, very good question. And I think it even touches on another question we've had on the chat about how you prioritize your portfolio given these constraints in some way. But David, perhaps you could start with that question.
Yeah, sure. So I mean, this is the topic we call capital allocation. Of course, which is what you're talking about. You started at an industry level, Syed, where we clearly have had over concentration in certain areas. In fact, some of the focus of today, oncology and rare diseases have had, perhaps, an outsized representation in running back almost a decade now, but certainly in the last 5 years. Now, given the capital markets that exist right now, in the so-called biotech winter, we're seeing much of that shake out. One of the questions that I think really is on the table is, will there be a rationalization of capital allocation across therapeutic areas? We made the decision that we would, for example, stay in cardiovascular disease at a time when, many companies were getting out, because the public health import, is enormous. I mean, in the last 2 years, far more people died of atherosclerotic cardiovascular disease than COVID-19, far, far more. Where are the NIH emergency committees? They have yet to appear. And so, this to me is something we grapple with on a daily basis. What should that capital allocation look like across therapeutic areas? Of course, it's driven by our scientific insights, the biology married with where's the greatest unmet medical need. To me, those are sort of the true north principles that you always come back to, and you have to sort of not then allow yourself to be distracted by what the herd might be doing. There are 600, give or take, who knows, CAR T companies now chasing actually a handful of targets, So the only thing we know for sure is that most of them, the large majority, will never return on investment. And so, that day of reckoning will come. This is not to depress anyone there, but to simply say we have to have a clear-eyed view of what's going on. And this is something that's top of mind right now.
Yeah, very, very good. Thank you, Syed. I'm going to, rather than go to other panels, we've got other people waiting, and I know we're approaching our end, but Karen, I think you're on. Karen Reeves, do you have a question for the panel?
Thank you. Yes, I do. Thank you very much, Martin, and thank you to everybody on the panel also. So it's a pleasure to be at this amazing conference today. My question has two parts. So looking towards the future, payers may have a bigger say on drug approvals and success post-approval. So how can we ensure value is baked in our clinical development programs? And number two, which regards advisory committee meetings, as most of you know, there have been all kinds of surprises and challenges with the advisory committee meetings in the past several years, actually. What can industry do to optimize that process? Thank you.
Thank you, Karen. Two great questions. Andy, I'm going to go to you for the first one. What can we do to bake in this value in terms of clinical trials? And then Mathai, I'm going to go to you for the second question on the advisory committee. So Andy.
Well, thank God, I was praying you wouldn't give me that second question.
I'm really looking forward to Mathai.
I thought Mathai's just the guy for the job there too.
Exactly.
So here's the lagging measure to your question, which is that if you get reimbursed in Europe, you've demonstrated value in your development program. That's of course not what you're asking, but that's a lagging measure, certainly. I think I'll go back to the theme that I think all of us are saying, which is when you go after something that is a very high unmet medical need and you have something that's differentiated, you don't need to do a lot to demonstrate value, right? When you're going after diseases that have no therapies or have really poor existing therapies, there's just very little that you have to do. I think the challenge that we run up against is that, most of what we do is in space where there are existing therapies, and in many parts of the world, in order to drive towards reimbursement, which is the end zone, right? Mathai, you said that the 50-yard line is approval, but the end zone is going to be reimbursement. You actually do have to demonstrate some sort of value proposition, and there's no easy answer because that's in conflict with something else that's very important to us, and that's speed, right? Because when you start to laden your clinical trials with endpoints that drive value demonstration, you oftentimes lose speed. I'll tell you our strategy. Go as fast as you can to get approved in the first major market, and then figure out ways beyond your clinical program using modern technologies, digital and other evidence generation approaches to really demonstrate that value for reimbursement.
Makes great sense, Andy. Mathai on the tricky second part of that question.
I have literally no idea how to answer that question. So the question is about how do you make the advisory committee process effective? And, you know, one glib answer is try to have a medicine that's so unequivocally amazing that you don't have an advisory committee meeting.
Yes.
And that we do, actually, that's more than half the time. And so, but honestly, it is an issue, and it's become more of an issue of late as conflict of interest rules become more and more stringent, and the advisory committees are very earnest, but sometimes the experience level isn't quite what we all want it to be. And so the companies need to tell their stories in obvious ways that caters both to regulators and the typically physicians on the advisory committee. There's no great answer other than full regulatory reform, where there's an equivalent that goes from a jury to an expert trio of judges, or the equivalent of that has expert review by objective third party, so achieves the same end, but is not an advisory committee meeting as we know it today. But short of that, it is around the companies, the sponsors, being crystal clear on the value proposition, and the questions that are kind of obviously will come up. And so if it's barely good enough as a medicine, and the FDA is looking for advice on it, and it's going to be challenging. It's going to be really challenging. There's no great answer to how it's going to be not challenging.
Actually,
I think you've given the perfect answer, Mathai. Please, Andy.
Well, no, I was going to say I like the process, actually, and I like your answer, because even if you have a transformative medicine, and even if you do have an advisory committee meeting, you're likely to get a positive response. And what I'm disappointed in is, if you look over the last year, there's been a precipitous drop at FDA in terms of the number of programs that have actually gone to an advisory committee. And in fact, we had a negative experience last year where we received a CRL after 8 months of back and forth after a missed PDUFA date. And I think had we had it's one of the feedback that we gave to FDA is that we want an advisory committee meeting, because we felt that we had a drug that was in a huge unmet medical need area, and we've gotten great feedback from the outside world. So I like the process. It can have some variability to it, but I think it provides a level of kind of objectivity that's not just the way the regulators might look at a disease.
I agree, Andy. I think Mathai's answer was great. It's incumbent upon us to come up with great medicines where it's much clearer, if possible. I know that's a hope here. Thank you for that couple of questions, Karen. So just in the last few minutes that we have, I'd like to just go to the panel for one quick round robin and just ask you all, what are you most looking forward to in the next year or two? And it can be new modalities, new medicines, whatever. Why don't you kick us off, Mathai?
So for gene therapy for fundamental disorders to really take hold, and even for diseases that are relatively common but have a clear genetic basis, it does work to clear out all the bone marrow and replace it maybe with the genetic basis that you want, or clear out the cells and replace it if possible. But the conditioning regimens today are horrible, and they're very difficult and challenging. So, I'm kind of excited to see what's possible in the coming few years, in the relatively near term, because there's quite a bit on the horizon on more benign ways of conditioning a person prior to gene therapy. And I think that will actually be transformational for a bunch of companies and bunch of diseases.
Thank you, Mathai. David?
Yeah, to me, continued advance in the development of what we call multispecific agents, PROTACs, or those that induce targeted protein degradation would be perhaps the simplest use case, but that is expanding to multiple targets, to multiple effectors. I think it's going to be an area that's as big as monoclonal antibodies became in the future. We're at the real beginning of that field. And to me, progress there should just be tremendously exciting in really making tractable targets that are currently undruggable.
Very clear. Thank you, David. And Andy, last words on your dreams.
We'll go back to a theme of this meeting, and that's India. And I think we're missing an opportunity to do what we all do our jobs for, which is to ultimately get innovative medicines to patients and access. And as we've said earlier, 1.2% of trials run globally are run in India. So we have a huge opportunity. As we heard earlier, it's the reasons that we're not running trials in India are primarily due to perception, not reality. So I see a great opportunity there in the coming year, Martin.
Great way to tie this panel up. Thank you, Andy.